Hello, everyone. Thank you for joining today's session. I'm delighted to introduce an important topic that underpins nearly every modern sequencing workflow: magnetic particles for next-generation sequencing, or NGS. Today, I'll walk you through how magnetic beads streamline DNA extraction, library preparation, PCR cleanup, and target enrichment, and how CD Bioparticles provides high-performance magnetic solutions for these critical steps. To begin, let's briefly revisit what next-generation sequencing is. NGS is a high-throughput sequencing technology capable of generating millions or even billions of DNA reads in parallel. It uses reversible fluorescent terminator chemistry, allowing DNA bases to be detected during synthesis. While NGS has outstanding throughput, the read length is limited to about 500 base pairs, which shapes the design of all upstream sample prep processes. In all these workflows, from extraction to library cleanup, magnetic particles have become the preferred choice, especially for automation-based and high-throughput sequencing pipelines. Now that we've reviewed the basic principles behind NGS technology, let's walk through the full sequencing workflow to see where magnetic particles fit into the process. This schematic summarizes the major steps. First, sequencing begins with sample collection, typically blood, saliva, tissue, or microbial specimens. Next, DNA extraction isolates genomic material from the sample. Once DNA is purified, the workflow moves into library preparation, where fragments are repaired, A-tailed, and ligated with sequencing adapters. In targeted sequencing workflows, we then perform bead-based capture and hybridization. Probe-bound magnetic particles enrich specific genomic regions of interest. Following capture, the prepared library enters the sequencing platform, where millions of fragments are read in parallel. After sequencing, bioinformatics processing aligns reads to a reference genome and identifies variants. Finally, results are translated into meaningful information through clinical interpretation and reporting. With the workflow in mind, we can now explore why magnetic particles are indispensable across multiple steps, from extraction to hybrid capture and library cleanup. Magnetic particles are widely adopted because they replace centrifugation with a magnetic separation step, which is much gentler, faster, and easily automated. They enable highly reproducible nucleic acid purification, minimize sample loss, and are fully compatible with robotic liquid handlers used in modern sequencing labs. For applications such as low-input DNA, single-cell RNA-seq, or high-complexity metagenomic samples, magnetic beads provide unmatched consistency and efficiency. To understand how they work, let's take a closer look at their structure. Most NGS-grade magnetic particles have a three-layer structure. At the center is a polystyrene core, providing rigidity and uniform size. This is coated with a ferric oxide magnetic layer, which allows rapid magnetic response and separation. Finally, the outer polymer shell contains functional groups, most commonly carboxyl groups, which enable strong and selective nucleic acid binding. This engineered structure ensures fast, reversible DNA binding and consistent performance across multiple purification cycles. Let's see how these particles function in the first critical step: DNA extraction. Magnetic beads enable pH-dependent DNA binding. At low pH, beads are positively charged and bind DNA efficiently. At high pH, the beads become negatively charged, releasing the DNA into solution. This method eliminates high-speed centrifugation and protects DNA from shearing. CD Bioparticles offers the Mag-ISO DNA and RNA magnetic silica particles, which provide high binding capacity and excellent recovery for genomic DNA, viral nucleic acids, and RNA. Once high-quality DNA is extracted, the next major step is library preparation. Library preparation transforms fragmented DNA into sequencing-ready molecules. This process includes end repair, A-tailing, adapter ligation, size selection, PCR amplification when needed, and finally, quality control. Magnetic particles are used repeatedly during library prep, most importantly during size selection and PCR cleanup, which are vital to achieving high-quality sequencing libraries. Let's examine each step more closely, beginning with end repair and A-tailing. DNA fragments generated by shotgun shearing have mixed ends. In this step, enzymes convert all fragments into uniform ends, and A-tailing prepares them for adapter ligation. Magnetic beads are essential here to clean up excess enzymes, salts, and reaction buffers. Our PCR cleanup magnetic particles provide high recovery of 200 to 800 BP fragments, making them ideal for preparing DNA for ligation. Next comes the addition of sequencing adapters. Adapters attach barcodes and flow cell binding sites to both ends of the DNA fragments. However, excess adapters or adapter dimers can severely compromise sequencing quality. Magnetic particles efficiently remove free adapters after ligation. Our NGS ligation cleanup beads are designed specifically to remove small adapter dimers and ensure a clean, high-quality library. After adapter ligation, the most crucial stage, size selection, takes place. Size selection using SPRI technology relies on polyethylene glycol, or PEG, to selectively bind DNA fragments of different sizes. Low PEG concentrations capture longer fragments, while higher PEG concentrations allow binding of shorter fragments. A dual-size selection workflow helps precisely isolate fragments in the ideal range, typically 300 to 500 base pairs for Illumina sequencing. CD Bioparticles offers NGS DNA select beads, engineered for reproducible fragment cutoffs and consistent bioinformatics results. Once fragments are selected, PCR amplification may be performed depending on the input and adapter type. PCR amplification is optional but often necessary for low-input DNA or certain library types. However, PCR introduces unwanted byproducts such as primer dimers, polymerase residues, and short fragments. A magnetic cleanup step removes these impurities and substantially improves downstream sequencing quality. Our high-fidelity PCR cleanup beads specifically target and eliminate small unwanted fragments while preserving the desired library molecules. Besides whole genome workflows, magnetic beads are indispensable in targeted sequencing applications. In targeted resequencing, biotinylated probes hybridize to specific genomic regions, forming probe DNA complexes. Streptavidin magnetic beads are then used to pull down these complexes, isolating only the regions of interest. After purification, the enriched DNA undergoes library preparation and sequencing. CD Bioparticles provides high-affinity streptavidin magnetic particles designed for hybrid capture workflows such as cancer panels, exome sequencing, and clinical diagnostics. To support all these workflows, SPRI technology plays a central role. SPRI, or solid-phase reversible immobilization, is the gold standard for size-selective nucleic acid purification. It offers high recovery, precise fragment control, and compatibility with both manual and automated workflows. CD Bioparticles provides a broad range of SPRI-based magnetic beads designed to deliver high yield and consistent performance in demanding NGS environments. Let's take a final look at the full range of our NGS-related magnetic particle products. CD Bioparticles offers a complete magnetic bead portfolio for the entire NGS workflow. DNA and RNA magnetic silica particles, PCR cleanup magnetic beads, NGS DNA select beads, SPRI magnetic particles for library construction, streptavidin magnetic particles for hybrid capture, and complete customization services for bead size, surface chemistry, and bulk manufacturing. Our mission is to provide reliable, high-performance magnetic solutions to support research, clinical sequencing, and industrial-scale workflows. To conclude, let me summarize today's key points. Magnetic particles are essential components in every major stage of the NGS workflow, from extraction to library preparation and target enrichment. SPRI-based magnetic purification provides speed, accuracy, and scalability, making it indispensable for both research and clinical sequencing. CD Bioparticles offers a comprehensive suite of magnetic particle solutions tailored to these applications, delivering exceptional performance and flexibility. Thank you very much for your attention. If you would like to learn more about our magnetic particle technologies or discuss custom manufacturing options, feel free to reach out to our team at CD Bioparticles.